Hello, and thank you for joining today's event with Calidi Biotherapeutics, which trades on the New York Stock Exchange American under the ticker CLDI. With us today, we have Eric Poma, Chief Executive Officer of Calidi. We will begin with a brief presentation in a moment, and then we will open the event to your questions. Welcome to everyone joining us today on X, YouTube, LinkedIn, and other social media platforms. To submit your question, we invite you to join us on Zoom. Use the link provided. Once in Zoom, click the Q&A button at the bottom of your window and type your question into the text box. Before we begin, please allow me to read the Safe Harbor statement. This call may contain forward-looking statements within the meaning of the Private Securities Litigation Reform Act of 1995. All statements pertaining to future financial and/or operating results, along with other statements about the future expectations, beliefs, goals, plans, or prospects expressed by management, constitute forward-looking statements. Any statements that are not historical fact should also be considered forward-looking statements. Of course, forward-looking statements involve risks and uncertainties. Eric, please go right ahead.
Thank you. Thanks, Craig, and thank you to RedChip for giving us this opportunity. I'm going to share some slides here and tell you the Calidi story, which I think is at a really nice inflection point. So, we at Calidi, we're developing what we think is the future for genetic medicines. I'm going to show you the initial data in oncology, and then at the end, talk about how we think this could extend beyond oncology. But what we're really doing here is delivering genetic medicines to distal sites of disease through systemic administration. We think this is groundbreaking and new, and we think this is a new path forward in oncology and other diseases. As you just heard, I'm going to make forward-looking statements here. But again, the idea here is, how do you deliver genetic medicine to, for instance, metastatic sites in a patient who has multiple metastatic sites that are not amenable to intratumoral delivery? That's what we're really doing at Calidi. I'll walk you through how we've carefully developed a RedTail platform to allow us to do this. RedTail platform allows for systemic administration so that the virus can go everywhere. It is protected from immune clearance through the envelope form, through CD55 expression, which I'll walk you through. It homes into tumor sites where it can lyse tumors and induce immune priming. And then we have this component of delivering a genetic medicine along with that. We're excited here because COGS look similar to a monoclonal antibody. So some of the most recent advances in oncology, like CAR Ts, have come with enormous price tags and huge difficulties around manufacturing. That's not the case here. We think manufacturing is straightforward, and again, COGS very similar to what you see with monoclonal antibody. Everything I'm telling you is data that's really kind of emerged in the last couple of years. We have broad IP coverage here, covering all of this. So again, really nice scenario where we have good COGS, good IP coverage, and something that we think is completely novel in oncology. And again, to ground us, I'm going to start with talking about oncolytic viruses. An oncolytic virus is just a virus that can replicate inside of a tumor. And why is this important? We've been looking at therapeutic development in oncology for about 80 years now, going back to Sidney Farber doing some of the first chemotherapy work. What we've seen here, and we all unfortunately have familial experience or friends experiencing this, is that patients may originally do really well on therapy, and then unfortunately the tumor comes back, and the tumor comes back resistant. And so what we've seen in oncology over this period of developing new therapeutics is that every time you develop a new modality, you may have substantial benefit in patients who have progressed on everything else, and that's what pushes the field forward. So you think about pioneering antibodies. Genentech and ImClone did this, pioneering monoclonal antibodies. You think about developing IMiDs for multiple myeloma. You think about CAR Ts for hematologic malignancies. Those represented new modalities where patients suddenly had potential to go into remission after failing all available therapies. People have been excited about oncolytic viruses because they can represent a new modality, right? They're destroying tumor through replication in a way that's very, very different than chemotherapy or signal transduction inhibitors, or immune-mediated effects. So there's been a lot of excitement around this. And there's some very nice proof of concept here. We saw Amgen get T-VEC approved a decade ago. More recently, CG Oncology is moving forward with a BLA filing for non-muscle invasive bladder cancer for their oncolytic virus. Replimune is at the FDA for their oncolytic virus in melanoma. Candel is filing on their prostate data. This is great proof of concept. You're showing patients who have progressed on all available therapies. If you deliver a virus to the tumor, you can get good activity. But the problem here is that this has been limited to patients where intratumoral administration is amenable. So these are patients who have tumor sites that are directly injectable into, that you can directly inject the virus into these tumor sites. And the problem is most patients do not fit that criteria. So the majority of patients will have metastatic sites throughout their body in sites that are not amenable to direct injection. So we're seeing with the agents I'm talking about here that if you can deliver oncolytic virus to tumor, and in this case, it's via intratumoral administration, you can have really good results. So that's great proof of concept that oncolytic viruses can represent a new modality. But the problem has always been now that it's very difficult to do this systemically. Instead, you have to do this intratumorally, and that limits the number of patients. And so the question is, why is that the case? Why is it very difficult to deliver oncolytic viruses systemically? If you're delivering a CAR T, if you're delivering a monoclonal antibody, if you're delivering chemotherapy, you're just getting it into the bloodstream. It's going and finding the tumor that way, and that's how you deliver things systemically. Why can't we do that with oncolytic viruses? Well, when you put an oncolytic virus into the bloodstream, your immune system does not know that this is therapy. Your immune system reads the oncolytic virus as an infecting virus, and it clears it. So we have this challenge of how do you avoid immune clearance so that you can deliver oncolytic viruses systemically. There's been some attempts here by just really pushing dose and injecting in huge amounts of oncolytic virus. Typically, the immune system will eliminate 90% of all oncolytic viruses that you put into the bloodstream. If you try to dose really, really highly, maybe you could overcome that. But what we've typically seen there is that you end up overactivating the immune system. You end up seeing a lot of toxicity. So Calidi's been working on this problem for 10 years. We've taken a different approach here. We don't think the solution is higher dosing. We think the solution here is how do you mask the virus from the immune system? And that's what I'm going to take you through today. We've created this enveloped form that expresses CD55, a vaccinia form that expresses CD55 that's enveloped. And everything we've seen in our animal models and our ex vivo human assays suggests that we can really avoid immune clearance here. We've designed and engineered the virus so that it only replicates in tumor cells and is really potently lytic in tumor cells. And then we've gone this extra step where we've added a genetic payload. So I'm going to take you through all of that here because, again, we think this is really groundbreaking. So just to start from the very beginning, again, we chose vaccinia virus as the basis for what we were going to engineer and improve on. Vaccinia virus has a lot of advantages. It's inherently tropic for tumor cells, so vaccinia virus does really well finding tumor cells and replicating in them. It's not like an adenoviral vector where it's receptor dependent. It also doesn't incorporate into host DNA, unlike lentiviruses, so you don't have the potential for transformation. So there's lots of nice facets about vaccinia virus to begin with. The first thing we did is we engineered out 3 key genes, thymidine kinase, viral growth factor, and A46R, toll-like receptor inhibitor, so that the virus cannot replicate in a normal cell. So this virus is inert when it sees normal cells, it can't replicate inside them. But interestingly, tumors have compensatory mutations for all those. So tumors are extraordinarily metabolically active. So thymidine is present. You don't need a kinase. VEGF growth factor is deleted here, but tumors are rich with growth factor, and tumors have already shut down toll-like receptor signaling. So again, you now have a virus, step 1, that can only replicate in a tumor cell. Again, vaccinia virus is great in terms of putting in genetic medicines because it's a very large genome. There's over 200 open reading frames, huge potential for inserting in additional genes. Calidi has identified some proprietary sites where you get good gene expression here. So that's been great. Vaccinia is also very, very good at expressing its genes. It has it's built in is the ability for it to shut down host host cell protein production in favor of viral production. So you have this vector that you can put in genes and they will be expressed extremely highly when the virus replicates. And again, this is now on a backbone where you only see replication in tumors. So the first two steps are basically saying we only replicate in tumors and we can put these genetic payloads in here that will only replicate and only be expressed in payloads. But the really big advance here was how do you avoid immune clearance, right? That's been the big question. How do you inject this into the bloodstream and avoid the immune system from clearing it so it can go and find these metastatic sites? And the way we've really done that is twofold. One, just the biology of vaccinia virus is such that in a typical infection, greater than 99% of the virus is what we call the IMV form, the intracellular mature virus. Less than 1% of the virus, though, is expressed as the extracellular envelope virus. What that means is that less than 1% of the virus, as it's leaving the infected cell, it takes on the membrane of that cell. It coats itself in a normal cell membrane. And you can imagine that by doing that, it becomes much harder for the immune system to recognize. It now looks, for all intents and purposes, it has the same covering as a normal cell. And so it becomes very difficult to identify that. We know this from just typical vaccinia virus life cycle. We know that the EEV form is very resistant to antibody neutralization, whereas the IMV form is not. We know that you can select certain cell lines where the virus, as it leaves and has those certain cell line membranes, is very protected from elimination. And we know that in a typical infection, the way you get dissemination during a typical vaccinia virus infection is because of the EEV form. That is what's able to enter the bloodstream, avoid clearance, and get to distal sites and propagate infection. So the insight Calidi had here was genetic engineering, strain selection to get a high EEV form, and then manufacturing that purifies out the EEV form. So this was a nice big step forward. We're creating a form of the virus that exists in nature, but we're being for the first time able to really create it as a drug product. That's the EEV form. But we went one step further here because we were concerned that the EEV form alone would not be sufficient. It's great in terms of avoiding immune clearance, but there's still some propensity for the immune system to be able to clear it. So we wanted to go one step further. What we did here is we engineered the virus to express CD55 in high levels through the envelope here. And the reason we did that is CD55 is a normal human protein. It's expressed on normal cells when you want those cells to avoid being cleared by the immune system. So biology here is very, very well understood how CD55 inhibits complement and immune clearance. For the first time, we've created a form of vaccinia virus that is enveloped and expresses high levels of CD55. So this is where we're really excited. We've presented lots of data at scientific congresses demonstrating that if you inject this virus into a normal animal, and again, CD55, it's a human protein, but it has the same effect in mammals. If you inject the virus into an animal with a normal immune system, they can no longer clear it, and you have very good efficacy. We've taken donor samples from patients, exposed those immune cells to the virus in a petri dish, and you don't get clearance. And so we're seeing for the first time by creating this EEV form with CD55 expression that you can really avoid immune clearance in all of our animal models, in all of our ex vivo human assays. So this is something new. This is something we're really excited about. This is the RedTail platform, a virus that only replicates in tumors, that can have genetic payloads, and that most importantly has the EEV and CD55 form that can avoid immune clearance. The first compound that we've created, the first virus that we've created, the first lead compound that we've created coming out of the RedTail platform, we call CLD401. Again, it's everything I described in terms of you're going to give this systemically. It doesn't get cleared by the immune system. It targets tumor cells. It lyses them. When it lyses them by replication, it primes the immune system. But we've also added this genetic medicine component where CLD401, as it's replicating in tumor cells, pumps out, expresses a ton of IL-15 superagonist. And again, IL-15 superagonist is a protein that drives the immune arm that recognizes tumor cells. So it drives activation of NK cells, gamma delta T cells, NKT cells. It drives an immune response specifically geared towards attacking cancer cells. So this is a 1-2 punch that we've created in the virus. Again, it avoids immune clearance, it gets to metastatic sites, it starts to destroy tumor by replicating, but it also starts to push out IL-15 superagonists into the tumor microenvironment, and that recruits NK cells, gamma delta T cells, all of that to come in, alter the tumor microenvironment, and now your immune system starts to destroy tumor through both an innate and an adaptive immune response. So again, nothing like this before. We're very, very excited about this. We expect to file the IND by the end of the year and start a phase 1 monotherapy study in solid tumor patients who have progressed on available therapies. We're looking at doing that study in the US. We're also looking at the potential to maybe start that earlier in Australia. But again, to just show you some of the data that we're really excited about. Why did we pick IL-15 superagonist? We know from the Anktiva experience that IL-15 superagonist, when it's in high concentrations locally, can really drive an immune response against tumors. So the history here is Immunity Bio has gotten Anktiva approved in bladder cancer, where you just directly inject extraordinarily high concentrations of Anktiva into the bladder. They're hitting concentrations of around 80,000 picomolar in the bladder because, again, you can just directly inject it all into the bladder. It stays contained in the bladder. And they've seen fantastic data for patients with non-muscle invasive bladder cancer. Drugs been approved for that. If you're familiar with the story, you know that they're also trying to go after patients with non-small cell lung disease that's metastatic. Here, they're injecting it into the veins, but because they're doing this systemic administration and because IL-15 activates immunity everywhere, they've had to dose at concentrations that are about 1,000 times lower than what you see in bladder cancer because you cannot activate the whole immune system everywhere in the body all at once. That's been the limitation. And again, the data in bladder cancer are phenomenal. The data in lung cancer, we're still waiting to see if these really low doses can be sufficient to induce an immune response. So if you keep that in mind, what we're showing here, this is an animal model where we have mice with a normal immune system with murine cancer. They have multiple sites of disease. We inject our CLD401 virus through a tail vein into the bloodstream here, systemic administration. Virus circulates, gets to tumor, and it only replicates in tumor. And at the tumor, you can see that we're inducing these huge concentrations of IL-15 superagonists. So you're at 60,000 picomolar, very similar to what you see with Anktiva and bladder cancer. But you're not seeing high concentrations anywhere else. The concentrations anywhere else are 1,000-fold lower. So you don't trigger any systemic toxicity where you're overstimulating the entire immune system. You're just locally stimulating the immune system at the site of tumor because the virus, even though you give it systemically, only replicates in tumor. And if you look at the data below that, this is if you have an animal with no tumor and you inject virus into it, and you inject CLD401 into that animal, you really don't see any IL-15 superagonist because, again, virus can only replicate in tumors. So we think this is a huge step forward where you're now able to deliver a virus systemically. It only goes and replicates in tumor. It only expresses its payload in tumor. And here, the payload IL-15 superagonist, we know, is a potent stimulator of immunity. And so what are we actually seeing in these mice where we're injecting the virus in these mice that have metastatic cancer? So this is an example that if you don't treat the mice, they all die very, very quickly. This is an extremely aggressive cancer in the mice. If you inject them with just our RedTail virus that doesn't express IL-15 superagonist, just the basic RedTail virus, you have some nice effects here. You can cure 10% of the animals. You can slow down tumor growth. But this is just the one punch of the virus getting to tumor and replicating and destroying the tumor cells via replication. But if you look at our lead compound, CLD401, which is the RedTail virus that also expresses IL-15 superagonist as a genetic medicine payload, typically we cure 70 to 80% of the animals here. We've done this multiple times. We're always curing somewhere around 70 to 80% of the animals because it's the 1-2 punch. Not only are you destroying tumor by the virus replicating, but IL-15 superagonist at high expression is driving a potent immune response against the tumor. So this is the piece that we're really excited about. This is what we think is really new. The idea that you could have a virus that withstands immune clearance, gets to tumor, induces the same effects that you see with CG Oncology's virus or Replimmune's virus or Candel's virus, but beyond just the replication, beyond just the oncolytic effect, it also has this extraordinarily potent IL-15 superagonist payload. This is what we're really excited about. And again, what's great here is that we expect to be in the clinic early next year and very quickly starting to prove this out. So we think right now I'm showing you, what we believe is extraordinarily compelling preclinical data, but within a year, I believe we're going to have clinical data. And if we are correct about this, we think the valuation increases here are going to be astounding. We continue to work on this platform. Again, what's really unique here is if you've built a platform where you can have a virus that can go anywhere you want it to in the body, you can target it by systemic administration, but target it to certain tissue types, and in this case, to cancer cells. What else can you do? So we can put multiple genetic payloads into this virus. So the first CLD401 has one payload of IL-15 superagonist. We've been playing around a lot with other payloads. One of the ones we're most excited about, and I'll spend a little bit of time on, is what if you could express T-cell engagers? And again, I'll walk you through that, but we think T-cell engagers are a great payload here. We've been looking again at additional payloads. We've also been starting to look outside of oncology. We started to see some preclinical data where we can target the virus to be replicating in activated B and T cells. So we can get some good replication in an activated B cell and an activated T cell. These are the mediators of autoimmune disease. So you now have a way of targeting B and T cells for oncolytic viral clearance, but at the same time, you could have a payload that is immunosuppressive. So you could get high expression of an immunosuppressive payload only at sites of disease. So again, if you know the autoimmune space, we've got some great drugs to shut down B and T cell activation, but when you give them systemically, you have all sorts of side effects, right? Because you're shutting down B and T cell activity everywhere instead of just at sites of autoimmune disease. Here we have a strategy potentially to shut down B and T cells that are activated, that are driving disease, and to have payloads that are immune quieting just at sites of disease. So this is, again, stuff you'll see us start to present data on. But to circle back to the TCE piece, which again, we think is really interesting. If you know the history of T-cell engagers, this is a molecule that can bind both a target like CD20 or BCMA on a cancer cell, as well as grab a passing CD8 T cell, grab it by the CD3 region of the CD8 T cell, and push those two together, and now the CD8 T cell will destroy the tumor cell. And again, if you know this story, this has worked extraordinarily well in hematologic malignancies. We've seen great efficacy against CD20 for non-Hodgkin's lymphoma. We've seen very nice efficacy against BCMA for multiple myeloma. We have not been able as a field to get this to work in solid tumors. And the reason for that there's probably three major reasons. One, in solid tumor, most of the targets exist outside of the tumor as well. So you look at a target like TROP2, it's expressed heavily on tumor cells, but it also has expression elsewhere in the body. And so you can unfortunately drive on target, but off tumor toxicity here. That's been a real limiting factor for development of TCEs. The other piece here is just you have limited concentrations. TCEs are antibodies, but they're dosed usually at a fraction of what you would dose an antibody, again, to avoid toxicity effects. So you just have low concentration here as well. And then the most important problem here, and the reason why this works in hematologic malignancies, but not in solid tumor. In solid tumor, hematologic malignancies, there's no tumor microenvironment. These are cancer cells that are floating throughout the body, and they haven't been able to surround themselves with a network that's immune excluding. But in solid tumor, these are tumors that have built a cage around themselves of immune cells and of fibrogen and all these other things to block immune cells from coming in. So you can have the TCE get into the tumor microenvironment, but there's no activated CD8 T cells for it to grab. Here, what we started to show with RedTail is that you can create a RedTail virus that expresses a TCE like a TROP2 T cell engager and expresses something like IL-15 superagonist that activates CD8 T cells. And so what we've been able to show here is when you do that, that RedTail virus will get to the tumor and will start expressing both high concentrations of the TCE, so high concentrations of a TROP2 TCE, very, very high concentrations, but again, only in the tumor microenvironment because it's not replicating anywhere else. And at the same time, it'll express high levels of something like IL-15 superagonist that will drive activated CD8s into the tumor microenvironment. And now you've met the conditions for TCE being active in the tumor microenvironment. So we've been extremely excited about this. We've been really moving forward with our lead compound for systemic administration in patients who have progressed on all available therapies. We've been seeing very excellent, unique data in the preclinical space there. And again, we expect to prove this out early next year. We are following on with things like this, T-cell engagers, and then we are following on with looking at autoimmune disease as well. So we think there's a huge potential here for the company to drive these programs forward, get recognition, get valuation increases based on what we're doing here. And I'll stop there. Happy to take any questions.
Thank you very much, Eric. Yes, we will take your questions right now. Press the Q&A button and type in your question. As we can take only your written questions today, we do ask that you please not use the raise hand button. We've already received several questions. Eric, can you walk us through the expected timing for your IND filing and what key milestones investors should be watching for as that process progresses?
Yeah, so we've indicated that we expect to file the IND by end of the year. We've been in discussions with the agency. We're very pleased with how everything's been going. So we expect to file the IND by the end of the year. And that would mean starting the clinical study early in 2027. And again, what's exciting to us is if I'm developing a new medicine for obesity, early anecdotal phase 1 data doesn't mean a lot. You need a huge data set to compare to what's out there. But in this case, if anecdotally in the first handful of patients, I'm seeing that we can inject this virus systemically, and you're seeing metastatic lesions in a non-small cell lung cancer patient or a head and neck cancer patient resolve, we've largely proven out that we've created a virus that can withstand immune clearance. So I can't stress this enough. We think there's a huge inflection point coming early in the phase 1. So that's one piece. You'll see us file the IND by end of the year. You'll see us announce first patient dosed first quarter of next year, hopefully. So those are two big pieces. But then throughout the year, we're going to be presenting at a host of scientific conferences as well as banking conferences as we update data and show additional utility, additional value in the platform.
Thanks, Eric. As we look ahead through the remainder of the year, what conferences or scientific meetings are you planned to potentially present data at?
Yeah, I mean, historically, this year we presented at AACR, we had some data at ASCO. We will present again at SITC, probably at ESMO, probably at the AACR Drug Development Conference. So there'll be a series of high-profile meetings that we will be presenting at.
From a regulatory perspective, what types of FDA interactions or feedback could represent important validation points for the platform this year?
Yeah, we've updated. We've had some type D interactions with the FDA that have gone very well for us. Those are usually geared towards making sure your CMC package is what the FDA is expecting. We will probably update on a pre-IND meeting as well here, and just give kind of some roadmaps that the IND is moving ahead well. So you could expect that you'll see us talk about pre-IND meeting as well in the near term.
I noticed your operating expenses in the first quarter of this year were close to $1 million less than the first quarter of last year. Can you expand on that?
Yeah, I joined the company about a year and a half ago, and myself and our CFO have worked really hard to make sure that every dollar spent here, we're getting the most bang for the buck. Before I joined, we were spending 60% of every dollar on G&A. We now spend basically 70% of every dollar on R&D. So we've just made a commitment here to be as lean and efficient as we possibly can. Capital, this is an environment where capital is not cheap. And so we're just making sure and we will continue to cut costs here and make sure that we are husbanding our capital and being very efficient with it.
As you prioritize capital allocation, how are you thinking about balancing pipeline advancement with maintaining runway flexibility?
Yeah, I think we are, we will be flexible where we need to be, but as I said earlier, as we get into the clinic, if we demonstrate that we have created a platform that avoids immune clearance, can get to distal sites of disease, express payload, we believe the valuation increases there will be huge, right? This would represent a new modality in oncology, just like Genentech represented a new modality and Celgene represented a new modality and Kite represented a new modality. So everything we are doing is in service of getting to that clinical um, inflection point as quickly as possible.
Looking across the business, what would you define as the most meaningful potential milestones for Calidi over the next 6 to 12 months?
Yeah, I think the IND acceptance will be a huge milestone. Again, we expect that to happen in the next 6 months or so. And then first patient dosed a few months after that, and then actual clinical data a few months after that. So we think that we are starting to accelerate the really meaningful milestones. When I joined the company a year and a half ago, a lot of it was heads down, get the manufacturing process to work, get lead selection done here, do the kind of behind the scenes blocking and tackling that we needed to do. Not a lot you can tell investors about our manufacturing process is getting better and all that, right? But now we are going to have some very, very visible milestones, FDA opening, opening of an IND, dosing a first patient, getting data in clinically. Those will be huge for the company, and I see those happening starting in the next 6 months or so.
And our final question for the day, Eric. RedTail has generated a lot of interest in oncology applications, but could the platform potentially have utility outside of cancer over the longer term?
Yeah, we think that the platform is well positioned if again, avoiding systemic clearance has been a huge problem for using viral vectors for gene delivery. So again, we think oncology is a huge space for us, but we're starting to look, as I mentioned, we're starting to look at autoimmune. We think that's another really large space where you need new mechanisms of action. And there's the potential to go beyond that as well. So we believe that this could really open the door for a huge amount of utility outside of just oncology.
Thank you very much, Eric, and thank you to everyone who submitted their questions. For more information about Calidi Biotherapeutics, reach us at 1-800-REDCHIP or email us at cldi@redchip.com. Please visit RedChip's investor information page for Calidi Biotherapeutics. It's cldiinfo.com. There, you can view and download the investor presentation and fact sheet and sign up for news alerts on Calidi. Thanks again to our many participants today, and thank you very much, Eric.
Thanks, Craig. Thank you, Richard.